Good afternoon, everyone. Welcome to third Presidential Session in CIB. My name is Pia Sormunen. I'm a Vice President of the CIB. I work also as an Associate Professor in Tampere University in Department of Civil Engineering in the Faculty of Built Environment. Today, we have a session called CIB W062, Water Supply and Drainage, Recent Advances in Legionella Research. And this webinar will provide a comprehensive overview of the activities of this working group, Water Supply and Drainage, which is particularly focusing on recent Legionella research. It aims to address a critical question. Is it possible to save energy without comprehensive health? It will begin with a brief introduction to Legionella, covering various species, then their growth conditions and preventive measures in the sanitary installations. Following this, it will dwell in the latest research for reducing domestic hot water temperatures while minimizing the risk of Legionella proliferation. This topic is crucial for reducing energy losses and enhancing the coefficient of the performance of heat pumps, which are also responsible for domestic hot water production. Our discussion will include an examination of both domestic hot water storage systems and those with instantaneous production. The webinar will conclude with an exploration of the ongoing and future research opportunities in this field. Today, we have here in Mr. Bart Bleys, who is the coordinator of CIB W062, Water Supply and Drainage, he has been a researcher at Buildwise in 2010, specializing in the building water supply and drainage systems. He has also served as a quality, safety, environment, risk and compliance manager and coordinated technical committee on sanitary and industrial plumbing, focusing on the topics such as water distribution, drainage, rainwater harvesting, non-potable reuse, domestic hot water performance, and hygiene design. His career began in the 2010 at the SECO and later PSSA, and he worked on the certification, inspection, and regulatory compliance for construction products, including sanitary and HVAC system. Binor machines. Okay, welcome, Mr. Bart Bleys. The floor is yours.
Thank you very much for your kind introduction. I'll share my slides. So I hope they are correct now. Thank you. So welcome again on this CIB President Seminar. I'm very happy to get this opportunity to discuss two of my favorite topics. They're really very important to me. And so the content of my presentation is, first of all, some general information about CIB Working Group 62, Water Supply and Drainage. Then I have an introduction on Legionella, the recent research on domestic hot water, and measures to prevent Legionella development, and then some conclusions and further research. But first of all, the CIB W062. So it's in pressure piping as well, so in the supply side, as well in gravity drainage, so the piping without mostly without pressure. And this topic also includes rainwater drainage. Then we have a lot of topics around dimensioning, so calculation and layout methodologies, very important to get a correct dimensioned installation. The next topic that we cover is material behavior, and more in particular, we have a lot of research on pathologies that can be found into sanitary installation, mainly due to corrosion. So if you have installation with different types of metals, they can be in contact or they can be further apart, but you will always get corrosion. And so this is one type of material behavior that we study. And a second large group is the use of plastic pipe materials. And everything involved in these kind of piping systems. We look at sizing and creating new installations, but we also have research on maintenance and renovation of existing installations. And one example of this renovation research that we have is if you have like an existing building and due to the desire to make to have water conservation, you change the toilets to have toilets with a lower flush volume, then of course you have to be sure that you will not create other problems like clogging in the installation. And we have a lot of research going on these low volume toilets, for instance. That brings us to the next point on the work program, which is the economy of resources. Water conservation is, of course, the main part, but also energy conservation. If you can reduce domestic hot water consumption, then you can also reduce the energy needed for heating up the water. So we are looking also into reusing water of different quality. Of course, for certain applications, you need drinking water quality, but for other applications, such as toilet flushing, for instance, you can use water of a lesser quality. You can use rainwater, for instance. You can recycle gray water that's coming from showers or bathtubs. After treatment, you can reuse them for toilet flushing. And due to climate change, these kind of topics on water conservation, they are becoming more and more important. We're also looking into environmental aspects. So water treatment, it's not always possible to have a centralized water treatment. So then you have an individual wastewater treatment system as a solution. And then we're looking into which of those have good performances and which of those are less performant. There are, of course, a lot of links to health and safety. And we're going to discuss this more in detail today. But we want that the water that is of a good quality when it's delivered by the water distributing company to the building. We want it to maintain this good quality throughout the building until the tapping point, so that there's no quality loss inside of the building, so that the user can have the same quality as distributed by the drinking water company. And so there are a lot of issues there. And one of them is Legionella. So we don't want Legionella development inside the installations in our buildings, mainly due to the health risks. There are also some countries working more on the testing sides, testing side of things, testing of piping installations and sanitary appliances. New product developments is also very big item on our work program, and we try to have a good collaboration with the industry. We even have presentations from companies themselves. They have the possibility to present technical papers on their innovations. And every year we have quite a few of those. Of course, all our topics are related to standards and regulations. So we also have more topics and advances in these kind of fields. And then the last point on our working program is also topics on Internet of Things, information and electronics and plumbing systems, like, for instance, leak detections, domotics, becoming more and more important also in this sector. So as you can see, it's a very broad range of topics that we discuss every year at our meetings. And well, it's very rich. If you come to the annual symposium, you get an update on all these topics. All the researchers working on these fields, they present every year their work on our annual symposium. And at the same time, it's a broad range of topics, but it's also a very kind of niche research field. So there are not so many researchers working on water supply and drainage. And it makes, for me, it's an advantage, because that makes our annual symposium very relevant. I think with our annual symposium, we're like in the top 3 on a global scale of big events on water supply and drainage. And we're very proud of it also. So that's a first a couple of slides on the work program and the topics that we have in W062. And as I said, we have an annual symposium, and it's not new. We've been doing this since quite some time now. I think it was actually the first one was in '74, so really a lot of years. We have the 51st symposium this year, so I think we missed 1 year due to COVID. And we are organized in 2 days of full days of technical sessions. So there are no parallel sessions. All is plenary, and we have 45 to 50 papers in one symposium, and that means about 15 minutes of presentation for each paper. We have a third day of technical visits and other visits. We have also a partner program if people want to bring their partner. And it's medium-sized symposium. We're about 100 plus participants of more or less 25 countries. Depends, it varies a bit from 1 year to another. And so afterwards, we produce proceedings, of course, of this symposium, which are published through the ICONDA database from the Fraunhofer Institute, and also on the CIB website. And you can find the link in the presentation. To accompany this annual symposium, we have a scientific committee, which is more or less distributed proportionally in the same countries as the participants. So you can see that we have on our scientific committee, we have Portugal, Taiwan, Slovenia, Brazil, Hong Kong, Slovakia, Japan, USA, and then again Japan, Taiwan, and the United Kingdom. So these are more or less also the continents that are participating. We have also from Oceania, so Australia and New Zealand, people coming to our symposium, and from from the northern and the southern part of the Americas. So it's kind of very global, our symposium, and it's very interesting also because the conditions in the different countries and the ways of construction are very different, and we can learn a lot from each other. Makes it very enriching experience, these meetings, these annual meetings. So last year, for instance, we had our symposium in Miami. It was the University of Miami and the Murphy Construction Program that we're hosting. Since COVID, basically, we have also a hybrid formula. So we have in person, but we also have the possibility for presenting online and following the symposium online. So that was 2025. In 2024, we had our meeting in Northampton in the UK. There, the host was the Aliexis group. So we try to have good context with the industry as well. And here it was even an industry partner hosting. That was the 49th International Symposium. And for instance, there was a technical visit of the National Lifting Tower that you can see on the right-hand side of the slide. And that's actually a large test tower for drainage systems. I think it's about 127 meters high. So you can really test drainage stacks for high-rise buildings. And in these kind of high-rise buildings, you even need pressure valves to compensate the pressure in the drainage system. So it's very interesting to see it also on a real size installation with transparent pipes, as you can see on the right-hand side of the slide. You can see actually how it works and how the water and the air flows through the pipes. So it's very interesting technical visit. And also a good example of collaboration between different countries, because in this test setup, a lot of research is being done by, for example, the Heriot-Watt University, but also by researchers from the United States, and other collaborations are ongoing. So it's very interesting, and it's one of the goals also of our working group to get this international collaboration even stronger. So the year before, it was in Leuven, Belgium. So Buildwise was hosting. You can see, like in the previous pictures, the group pictures and the setup of the symposium room. We often have it in a hotel because the group size allows for it, and it's quite practical to have the meeting venue inside of the hotel where most people are staying. And then we did the technical visits of the Buildwise labs, and of course there's always some dinners in the evening, a gala dinner as well. We have a tradition even of karaoke for people who like those kind of things. So it's also a lot of people coming back year after year, and it's like one big family, and it makes it also a very nice 3 days where a lot of people are looking forward to it. And then, of course, we're already planning for next year. And so next year it will be in Maribor, Slovenia. The call for papers is already open, and you can find the deadlines on the left-hand side of the slide. And it will be the University of Maribor that will be hosting this time. You can introduce abstract till the 24th of March. And again, it's very it's one of my favorite topics. Is one of my favorites 3 days of the year. So uh, I think once people come to the symposium, they keep coming back, and I think that says a lot. So um, that was the first part of the presentation. So W62. Second topic that is also really one of my favorites is Legionella. And as Pierre explained, we have been working now on some time on the question, is it possible to save energy without compromising health? And why are we working on this? Because first of all, Buildwise was founded by the Belgian building contractors, so we have to reply all their questions. And one of their questions that we often got was, okay, we can produce domestic hot water at 50, 55, 60 degrees. Um, the there's sometimes the domestic hot water production unit, the water tank has an anti-Legionella module. At what temperature should we program it? Because often the manufacturer says it's programmable, but they don't say at which temperature it should be programmed. So that was our starting point. We also um, wanted to see whether it was possible to reduce the domestic hot water temperature because of energy savings. In Belgium, we have a lot of Combilus installations, like you can see in the middle of the slide. This is a type of installation where we have the heating, so the energy is centralized, but it's distributed in a two-pipe system to the different apartments in a building, for instance, and then decentralized. So it's technical water that's being distributed, and decentralized. There is the domestic hot water that's being produced, so that avoids more pipes for distributing the hot water when it's also produced centralized. Here is a decentralized production of domestic hot water. So there's only cold water distributed to the apartments. And often these satellite units, they work on the heating side with underfloor heating. And so these are like small district heating networks inside the building. And of course, if you have underfloor heating, then low temperature is sufficient. And you have to only have these higher temperatures for the domestic hot water if you want to produce it at 60 degrees. So that's a lot of effort to that's that imposes higher temperatures on the complete system. And then you get the question, is it really necessary? So that was one of the reasons also. We had to installers questions, and then we have some pressure on these high temperatures because of the low temperature district heating networks, and also because of um, heat pump domestic hot water production through heat pump boilers, as you can see on the right hand side. Of course, the COP of such an installation is a lot better if it can produce at 50 or 55 degrees instead of 60 or 65. So these three reasons are there for looking into lower temperatures. But of course, for us, it's important to do it without increasing the health risk. And that's what we were looking into. Do we have to choose one or another, or can we find a solution that combines both and that makes sure that you don't have to choose? So just a brief introduction on Legionella. So there are different kinds of Legionella. There's a Legionella pneumophila, which is pathogen, which is the cause of almost 90% of the people getting sick. And then there's other Legionella. Legionella species is the group name, and inside of the group name you have some pathogenic, Bosmani, Anissa, etc., and then some non-pathogenic. And the species they grow better in low temperatures and colder water, and the pneumophila better in high temperatures. And so the first time we really actually got aware of the risks was in 1976, where there was in Philadelphia a large group of people that got sick. It was a group of legionnaires. So that's where the Legionnaire's disease got its name. And so in the middle of the slide, you can see the bacteria on the first picture, and the second picture below. That's how they are cultured in the lab to determine the concentration. So you take a water sample, you put it on a petri dish, and every bacteria becomes a white dot on the plate, and then you can recalculate the concentration. So how do you get sick from Legionella? Because Legionella is a bacteria that is literally almost everywhere in water, almost every type of water, and in soil. Uh, Only in salt water there's no Legionella, but for the rest, Legionella everywhere. There's even Legionella in drinking water, not much, one bacteria per cubic meter, but still in the water distributed to the building, there is Legionella. To get Legionellosis, so a lung infection, you need to inhale small droplets of water that contain Legionella, and they have to go really deep into the lungs, and then they can develop and create the Legionellosis. So to get the disease, you have to have Legionella in the water. It has to attain a certain concentration. Then it has to be in an installation that creates aerosols, the small droplets. You have to inhale them, and then if you have a person um, uh, that's a bit older or that's recovering from cancer, for instance, they can develop the Legionellosis. So that's how it works. There is no contamination from person to person. So if you get a Legionellosis, you cannot contaminate other persons. So patients are not placed into isolation. There's no contamination via hand-to-hand, no vaccine. So there's some similarities to the COVID, but it's not contagious. You have to be in contact yourself directly with the source. There's also no science-based risk concentration, but generally accepted is the Legionella pneumophila concentration below 1,000 has a negligible risk, except for the high-risk patients, as I said. They're better to have even lower concentrations in the water. If you look at people that are fragile or that are more likely to develop Legionellosis, that's very similar to COVID-19 actually. So older persons are more sensible to get it, and you're very quickly older, even just above 50 is already old for Legionella development. Men are also more sensitive than women, so those two combined, the most infections are in men even over 65 years, as you can see, of age, as you can see in the graph on the right-hand side. The picture above is a picture of a person with a lung infection with Legionellosis, which is also quite similar to pictures of people having COVID. And the same other risk factors apply. So if you have people, smokers, alcohol consumption, people with a chronic illness, people with a weakened immune system, recently undergone surgery, etc. So the same kind of factors, risk factors, as in COVID. If there are also two types of disease that you can get. You can either get, if you get sick, of course, you can either get a complete Legionnaire's disease, or you get Pontiac fever. It's not the same thing. Legionnaire disease is a lung infection. You have a long incubation time, 2 to 19 days. Pontiac fever is much quicker, 1 or 2 days after being in contact. The symptoms are also different. In Legionnaire's disease, you have fever, chills, cough, So more symptoms that are often firstly diagnosed as pneumonia, and in Pontiac fever, it's just fever. Um, and then, of course, there's the big difference in mortality. If you get Legionnaire's disease, there's an average mortality of 10%. So that's young and old people mixed, sick and healthy people mixed. So 10%, it's not nothing. And if you go to people that are more fragile, then it's 20 up to 40%. And of course, Pontiac fever, there is no mortality. So these are the two types of disease that you can develop. So, of course, we do not want this. And then you have, and you know that at the same time, Legionella is coming into our buildings through the drinking water. So then you have to understand the growth conditions. So the basic growth condition is temperature. So if you can keep your hot water hot, and that means at least 55 degrees, and your cold water cold, because you can also have these other type of Legionellas, the non-pneumophilas, that develop in the cold water, then development inside of the installation in the building is limited. You do not get high concentration. That's important. Temperature is the main thing. Second factor is stagnation. Water should flow in the installation, and you should not have a lot of dead legs where a lot water is stagnating. So at least all the water in all the pipes should be renewed weekly. So it's also um, a factor in holiday periods in schools, for instance, in holiday rooms that are not being rented permanently, etc. So temperature, stagnation, then you have nutrients. You have to have food for the bacteria, also oxygen. It's very important. And then they can develop better if there's a sort of a habitat. If you have some sludge, corrosion products, biofilm, then they are very happy and protected, and they're also a bit more protected from the temperature. So there are differences between temperature in lab and temperature in real sanitary installation. The results, you need some higher temperature to compensate for these kind of habitats where they are protected and they can develop more freely. So the number of cases, are there a lot of cases? Is it a big problem or not? In the graph here on the right hand side, you can see the number of people getting Legionellosis per 100,000 persons. The more dark brown you get, the more people get Legionellosis. So the darkest brown, like in Spain, is 4 people out of 100 persons get Legionellosis in this year that was reported. So this is the graph for 2022. For the whole of Europe, we had 11,447 cases reported, because once there's a Legionellosis detected, it's mandatory to report it. So we can identify if there's a cluster and if you should look for an active source or not. And from these 11,447 cases, 801 person fortunately died. And for 80% in this year, they were confirmed to be Legionella pneumophila and not the species, so the hot water variant, the hot water species. Okay, so quite a lot. Of course, if you compare it to traffic accident numbers, it's not so much. But if you compare it to building related deaths, it's often the second or the third cause, first being fire, and then it's a bit in competition with the carbon monoxide. That's in some countries, carbon monoxide is more important. In other countries, Legionella is the second cause of that's related to buildings. And you can, in the sick person, take samples. You can, in an installation, take samples, and you can match them on a DNA basis. So you can, from a liability point of view, you can identify the cause. One more graph on the USA. You can see a bit less. They have a bit more chlorine also in their water, which is a factor. But I'm showing this graph because there's also an increase over the years, not only in the US, but generally in all countries. You can see that the incidence of Legionella is increasing, and we think it's partly due to the use of lower temperatures for domestic hot water. So a lot of good reasons to have research. And we wanted to see, okay, can we not reduce the domestic hot water temperature to, like, for instance, 45 degrees? We know it's not good for Legionella, but we want to compensate the risk by having thermal shocks. And we want to have thermal shocks, the smallest possible, that is still effective on the Legionella side, to combine energy savings and Legionella safety. That was the setup of our first research. And so, of course, important note for us, at least, hygienic water quality health is more important than energy savings, but if possible, we want to have both. And we did this in our lab. We have a large scale test setup with 40 meters of circulation pipe, a domestic hot water tank of 200 liters, a culture tank. So it was a reserve if we found a solution that we could recontaminate the complete installation. So this is really quite unique. Actually, there are not so many research centers that can build a complete hydraulic domestic hot water system and that are allowed to put in real life Legionella pneumophila, the dangerous kind, at very high concentrations. So it's very, very unique what we can do there. And the idea was, okay, we will find a shock, it will be effective, all Legionella will be dead, and then we can recontaminate from our culture tank. So that's what was the idea. A sneak preview, we didn't need to use our culture tank at all because we didn't find a lot of thermal shocks that got completely rid of the Legionella. What's also important to say, so we have a 200 liter domestic hot water tank. We have 2 tapping pipes. On the 7th floor of our tower, we have a shower profile, and on the 1st floor, we have a kitchen profile. These profiles, shower profile is longer tappings at a higher flow rate. Kitchen is smaller tappings at a lower small rate. And I insist on this because you will see it in the results. Some other thing I have to stress is, so we contaminated the installation initially, and then we stopped adding Legionella into the system. So we had biofilm everywhere, and we stopped adding Legionella. We just started adding clean drinking water. So we start from a contaminated installation, but we do not continuously add Legionella. And then here on the test tank, you can see a destratification, so an extra pipe and pump, and that's important to get higher temperatures while we are doing a shock, also at the bottom of the tank, because there's a lot of Legionella developing there. So it's very important also to have this kind of destratification pipe. Okay, yeah, so this I think I've already said. So that we had very high concentrations at the beginning, between 100,000 and 1 million CFU per liter. So remember the health risk limit was 1,000. So we have a factor of at least 1,000 times higher, because if you work with microbiology, you need to reduce at least 2 log factors to have a real effect. So we have to start really high to measure an effect. And yeah, the tapping profile is about 165 liters a day, which is of a representative of a single family house. The 200 liter tank is so as well. And so we started to producing at 45 degrees in a contaminated system, getting clean drinking water, and then looking for the shock that was effective. Here's a bit more detail on the tapping profile, and you can see that the showers, we have a larger tapping duration, 360, 400 seconds. In the kitchen, there are smaller tappings, 6, 20, 30 seconds. It's important to understand the results. And then we started, first of all, with a small shock, so one times a week, 60 degrees, 30 minutes. That wasn't enough. So then we went to 1 hour, wasn't enough. Then we said, oh yes, but we forgot to put on this extra circulation on the tank. We added this, still not working. So we started disinfecting the tapping pipes at the same time of the thermal shock, which makes sense, but still not working. Then we went to 7 shocks a week at 60 degrees, still not working. So, okay, at that moment, we started increasing the temperature of the shock to 65. We started over at 30 minutes, one time a week. Not enough, 1 hour, not enough. Extra circulation on the tank, because this extra circulation is typically for Belgium. Never seen it anywhere else in the world. It's quite simple measure, but we tried it without because otherwise it's only a Belgian solution. So we added the disinfection of the tapping pipes, and then for the first time we found something that was okay, that was working. So good news. But it's not very practical. So you needed to heat up to 65 degrees for 1 hour and then open all taps. In our installation, it's quite simple. We have 2 taps, but if you have to do it in a hospital or a nursing home, it's not so practical. So then we went to increasing the heating duration to 24 hours to use all the normal tappings that normally occur in the installation, and then to identify the taps that needed to be opened extra. So for instance, the showers, they are used long enough to disinfect themselves if the shock is long enough here. But the kitchen, you needed to go and open the kitchen faucet because there the normal use time was long enough. That's the main conclusion. Okay, and so if you put it in a graph, you can see this horribly complicated graph. So you have Legionella concentration on a logarithmic scale. The green pluses are the moments where we did a thermal shock. Each point is a Legionella concentration. The red ones are in the departure of the loop, orange return of the loop, blue shower, and yellow kitchen. This red line is 1000. This is the goal to be below and the Belgian limit concentration. And you have 2 main zones in this graph. You have the 60 degrees and the 65. You can immediately see that at 60 degrees, nothing really worked. We sometimes went a bit below, but it immediately after shoot up again. So main conclusion, 60 degrees is not okay. 65 was much better. So the first time it worked was here. All the colors are below, and that was where we had 1 hour of 65 degrees, and you had to manually open all the taps. So working, but not practical. Then we went to the 24-hour system where we used the normal usage of the installations, and you can see that only the kitchen, so the yellow dots, are above 1000. So then we went on to increasing the tap duration of the kitchen, so from 30 to 60 to 90 seconds. And then here at the first time, we had a solution. So it was about here. Everything was nicely below. Then we stopped doing shocks, no green pluses here above. And you can see that immediately it shoots up again. And then we reproduced this the same 24-hour shock with the same duration in the kitchen, and it worked again. So that's 2 repetitions, and we did 3 more to be sure. So this is the main result of this research. Then we had a second research with the plate heat exchanger. And so a lot of people in the sector are thinking that domestic Legionella is a storage problem. Well, with the plate heat exchanger, we have also, we did the same kind of research because there's no storage. And then we get directly to the results because I'm aware that time is running out. So the same graph, we did a plate heat exchanger working, producing at 45 degrees. First with different flush rates because some theories said that because the distance between the plates is so small that the velocity will be high and biofilm will flush automatically. We definitely did not see that. So different flow rates didn't flush Legionella at all. And then we implemented shocks at 60 and 65, and even at 65, it didn't really work. But if we put the plate heat exchangers continuously at 60, then you can see here in green that it was much, much better. And we continued even a bit longer than in the graph here. And it went nicely below 1,000, as the legislation in Belgium says the heat exchanger should be at 60 permanently, and then even a contaminated installation goes right down below. So there's a good reason for the high temperatures. It's possible to manage it with heat shocks, but it's not easy, and we actually only found a real solution in systems with storage tanks and not so much in systems with heat exchanger. So this was first results of thermal shocks. Other ways of reducing Legionella concentration is with dosing chemicals at the building level. It's permitted in some countries. It's not permitted in all German-speaking countries, so Germany, Switzerland, etc. In generally, it's not what we recommend because drinking water is a food basically, and we don't want to dose chemicals into it. So then the third solution that we're looking into now is non-chemical techniques, alternative techniques of reducing Legionella concentration in a circulation system. And then we're talking about UVC and ultrafiltration, so non-chemical solutions. And so we have actually a project going on in Belgium, which is called Optidis, still running until 2026, where we, amongst other things, because main goal of the project is to optimize circulation systems as a whole, and Legionella is one work package of this project. And we are doing this together with Kiwa Water Research, so a Dutch research center, because we discovered through CIB, and that's actually one of the added values of our symposium, is that we discovered that we had overlapping research projects in Belgium and in the Netherlands. And then we got together and we said, well, we can work together. If you test this, then we test the other part. We exchange the results, and we have both countries have more results, and we have a nice collaboration thanks to CIB going on this subject at the moment. So between the Belgian project, Optidis, in this project, we also are creating a second test setup where you can see some pictures on the right-hand side. And the second project in the Netherlands is a reload from Kiwa Water Research. They're also working on circulation systems. They also have a working package on non-chemical alternative techniques. And as you can see on this slide, they are doing the tests also on our first test setup. So it's a very, very nice collaboration between the two research projects, between the two countries, and all thanks to CIB. So I just want to repeat a warm invitation for the next symposium in Maribor. So it will be conference dates 18 to 20th of August. Thank you very much for your attention. And if you have any questions, you're very welcome.
Yes, thank you very much, Mr. Bleys. Thank you. Very interesting presentation. And okay, do you have any questions online? You may just open your microphone and just ask a question. Okay, no one, but I have a question for you.
Okay,
I'm
thank you.
I'm interested about heat pumps and
Yes.
how the heat pump technology is taking care of the Legionella and how the risk can be prevented in
Yeah.
heat pumps.
Yeah, I think it's not so very complicated, actually. It's a very good
Mm,
question. A lot of thermodynamic domestic hot water units, they also have an electrical resistance built
yeah,
into it.
yes.
And that can be used to do a thermal shock.
Mm, yeah.
And it can even be combined with photovoltaic electricity,
Mm,
so that you can do the shock when there's green electricity available. And that's also one of the interesting parts of our research. We can have like input of a frequency for the controller of this combination of PV
mm,
with the electrical resistance to have to disinfect at least the hot water production when there's a lot of sun, for instance.
yeah.
And I think that's not so complicated.
Yeah, okay, that's good because in my house we have a heat pump, and of course I think it's quite common technology nowadays
Yes,
in the
yes,
Nordic countries to heat houses. Okay, this is my understanding also to use in this kind of heat shocks, so to prevent Legionella. Okay, I have another question. What do you think? What are the risky building types what we have in Europe?
well, the more complex or the bigger the installation, the higher the risk that there are some parts of the installation that are suboptimal, or there are mistakes
Mm,
or pipes that are not being used.
yeah,
So if there's an increase of risk with the size,
yeah,
and the second risk is not linked to the building type, but to the type of persons inside the building. And you can see that
mm,
a lot of legislation in Belgium, but not only in most countries, and the Drinking Water Directive also asks each member state to define which installations are higher risks. And
yeah,
so that are mainly linked to the persons inside of the building. So the hospitals where you have
mm,
sick people, or with the immune systems that's reduced, or the nursing homes where you have elderly
yeah.
people. So it's basically linked to the size of the installation and the type of, if there are fragile people living in the building
Mm.
or not.
Okay, this is interesting. I didn't think that in that way. Okay, and okay, is there any questions online? Maybe not. So maybe you have your colleagues from the working group online listening you,
It's
knowing
possible, yeah.
yes, knowing everything. Okay. If we don't have any questions, so I think thank you very much, Mr. Place, and it was our pleasure to hear your presentations and about your research. It's very important, especially when we consider we need to reduce energy consumptions in the buildings, but in the same time provide health environment for people to live in. Okay, thank you very much. Thank
Thank
you.
you.
Thank you all participants. Okay, thank you and bye.